particular virus, but viruses are a particularly interesting group of organisms because they are pegged to be the most diverse and the most abundant organisms on the planet. So, to put this in perspective, there are 10 to the 23 bacteria in the global ocean. There's 10 to the 24 viruses in the global ocean. So they are really, really abundant. Just to further underscore this, viruses, there's estimated to be about approximately 1,000 viruses per every individual species on the planet. So just imagine if there's 2 million species, there's likely 200 million species of viruses out there. But with some basic definitions of a virus, it's a non-cellular particle that infects the host cell where it reproduces. The viral particle, or this is also called a viron, is essentially an infected nucleic acid. This can be DNA or RNA, and it's simply contained within a protective shell made of protein. And this protective shell of protein is called a capsid. Viruses have likely evolved from multiple sources, including some bacteria through degenerative evolution. And there's three current hypotheses for how viruses came to be. So the first one is the virus early process, where you had a pool of replicons, aka things that are capable to replicate on their own. They were encapsulated in some protein, and over evolutionary time, they increased in the complexity and through the emergence of translation to then become infectious. There's the regression process, where there was either a protocell or a primitive bacteria or archaea or even a eukaryote, and it underwent reductive evolution, and this was due to being a parasite, so it was a parasitic organism, and because when you're a parasite, you shed all sorts of genes, and so as it shed more and more genes, it transitioned to becoming a virus. And then finally, there's the escaped gene hypothesis, where there's essentially something very similar to the early virus hypothesis, where a gene that or a set of genes escaped and was capable of autonomous replication. This sort of autonomous replication we know occurred early on in life, as we talked about in the origins of life class, and we do know that viruses they can I'm not sorry virus pieces of DNA and RNA have the capacity to replicate autonomously. So, in terms of all of these, we believe that the regression hypothesis is the most likely, followed by the virus early process, and then finally the escape gene hypothesis. The viruses, again, are the most abundant biological entities on the planet. There's about 10 to the 31, which is a lot. And so we could ask the question, if you line them up end to end, how far would they extend? And the answer to that is 100 million light years. To put that in perspective, our galaxy is not even 100 million light years wide. So viruses are extremely abundant. The next question we can ask is, given how abundant they are and that they're light years of long of viruses, how are we all not dead? Well, the simple answer is that most viruses infect bacteria. And bacteria, again, are outside of viruses, are the most abundant life forms on the planet. So the vast majority of viruses aren't things like HIV or COVID-19. They are bacteriophages. And a bacteriophage or a phage is simply a virus that infects bacteria. Viruses are pretty simple. They have nucleic acid genomes. So we think of most living organisms on the planet having a double-stranded or dsDNA genome. However, viruses have double-stranded DNA, single-stranded DNA, double-stranded RNA, and single-stranded RNA. So they can have a particularly diverse way they package their genome. The virus genome is surrounded by a protein coat or a capsid, and this protects the protein from harm. And in addition, viruses are pretty variable in sizes. They range from 20 to 800 nanometers in size, and this is typically determined by their genome size. So viruses that have bigger genomes are typically larger in size. Viruses generally have four basic morphologies, and there are many exceptions. Unlike their bacterial counterparts, viruses take an immense shape of sizes and structures. But the four basic morphologies here are capsid, making a helical structure, capsid forming a geometric shape with flat sides. So the most common one we see is isohedron, which is a 20-sided thing of protein. The next one up is virus can get coated in the host lipid membrane. This is what we call envelope viruses, and complex viruses with extra shapes in addition to the capsid. And this just to show you some of the shapes we commonly see. So this is what we're seeing is a T4 bacteriophage. This is the most common shape for phages viruses that infect bacteria. It looks sort of like a lunar lander. This is a encapsulated virus, a Rehabovirus. And then we can see the octahedron and decahedron shapes. Viruses take a vast diversity of shapes. And so, for instance, Ebola looks very much like a worm. Um, the flu looks very much like this. So viruses have a pretty immense diversity of anatomy. But the virus structure is basically sort of differentiated by whether the viruses are naked or enveloped. And a naked virus simply just has these capsomeres surrounding its nucleic acid, and it's a very, very simple structure. Whereas an envelope virus has that capsid, and that capsid is then surrounded by a protein envelope. Um, a mature form of a naked virus has no parts of the host cell, whereas many of those. Sorry if you can hear my dog crying. She's looking for love. Sorry. The mature form of a naked virus has no parts of the host cell associated with it. It only has genetic material and a protein shell. Um, and an envelope virus structure is derived. The envelope itself is derived from the host cell membrane during exocytosis. Exocytosis is essentially when the virus breaks out of the host. And essentially, when we're looking at an envelope virus, this envelope virus derives from the physical host itself. The envelope virus structure has a lipid membrane surrounding the capsid. This is what we call an envelope. The envelope does come from the host, as we mentioned, but it is not encoded by a viral genome. There are proteins embedded in the envelope that are coded either by the virus or the host. It simply depends. It coats, remember that inner virid capsule, and this capsid itself is actually encoded by the viral genome. It does, as this is sort of exiting the host cell, as we'll talk about in a moment, it fuses with the host cell membrane during subsequent infection. And so viruses are classified by a much more sort of primitive way than we do, say, with bacteria or archaea or even eukaryotes. They don't have any sort of common genes like the 16S ribosomal RNA gene. Their genomes are a bit hard to use to classify them because they're generally extremely small, being on average, like say 9,000 kilobases or nine kilobases or 9,000 bases long. They're very diverse, and they don't have enough conserved genes. Even within a given family of viruses, there's a vast diversity of the genome. So you can just put that in perspective. You can think of something like the flu, all the different strains of the flu virus. It has huge, immense diversity of the genome itself. But viruses are classified based on a number of different characteristics, principally the shape and size of the viruses, simply because the more closely a virus is related to one another, the more similar they are in the characteristics. And this is a bit different than bacteria, very similar to the way we characterize eukaryotes. In some cases, the host range of the virus and its mode of spread, i.e. being an insect vector virus, allows a little bit better classification, simply because things that are closely related generally infect the same host. And so just to put that in perspective, that's like something like the flu, or comparing SARS with, say, COVID-19, which is the current outbreak. They are sometimes classified by their genome structure, whether they're DNA, RNA, double-stranded, single-stranded, and so on. But viruses are organized into families, genera, and species, very much like we are, very much like any other species on the planet is. The family name always ends in viridae, and the genus and species names always end in virus. And so for influenza, for example, the family is the Orthomycoviridae. I probably didn't say that right, but I'm terrible at pronunciation. But the best way we know to classify viruses right now is actually Baltimore classifications. It's based upon genome structure. And so the way this breaks down is we have group 1, which is double-stranded DNA. An example of this is the herpes virus. The genes can be just transcribed directly by standard RNA polymerase. It's typically from the host cell, but it can also be from the viral genome. Group 2 viruses are single-stranded DNA. These are typically a group of viruses that infect plants. So the Gemini viruses, they infect tomato plants and other agricultural exports. We don't particularly see group 2 viruses infecting humans or anything related to humans. It's particularly they focus on plants. However, these group 2 viruses do have single-stranded DNA, and they require the host DNA polymerase to generate a complementary DNA strand. So they take the template here, they use the host polymerase to generate a double-stranded DNA. Once they generate that double-stranded DNA, they then go on to infect the host. Group 3 is the double-stranded RNA. And so this is the rotavirus. So for instance, many of you have children. You might have heard of rotavirus because it causes pretty severe diarrhea in infants. But group 3 is a double-stranded RNA, so they require a viral RNA-dependent RNA polymerase to generate mRNA, and they basically use their double-stranded RNA to make a piece of mRNA to then go and attack the host. Group 4 is the plus sense single-stranded RNA. Plus sense is simply the top strand of the DNA itself. The single-sense RNA, it's best known as the poliovirus, but this is also where COVID-19 falls, so our current pandemic. These consist of a positive sense strand of RNA. This is the top strand, this is the coding strand of RNA, and this basically serves directly as messenger RNA to be translated into viral proteins once inside the host. Group 5 is our negative sense RNA viruses. This is where influenza falls in. They have genomes that consist of a template or a negative sense RNA. Thus, they have to package their viral RNA-dependent RNA polymerase for transcribing the minus sense RNA to plus sense mRNA. So basically, they have the lower strand, if you think about it, on a piece of DNA, and they have an enzyme that essentially translates that into the top strand, which is then the coding strand, which then can be used to infect the host. And then finally, we have group 6, which is the retrovirus. This is where HIV falls into. We're going to talk about HIV a bit more when we talk about viral evolution, because it is one of the most heavily studied viruses in terms of this family of group 6. They have no RNA polymerase inside them. However, they package an enzyme called reverse transcriptase. And what this does is it transcribes the RNA into double-stranded DNA. And then the double-stranded DNA is then used to infect the host. We're going to talk about this enzyme, reverse transcriptase, later on in the class. It's a particularly important thing to understand why HIV is so infectious. And then last but not least is group 7 with the pararetroviruses. This is hepatitis B. They have DNA, which is then transcribed to RNA, which is then reverse transcribed to DNA using its own reverse transcriptase, or one provided by the host. Sort of a weird group of viruses that we're not going to talk about very much. And this is just sort of a visual depiction of how things go. But ultimately, all these things have to be transcribed to RNA, messenger RNA in some way or form. Stickers, lie down. But how it gets there is a pretty distinct way. It has a pretty distinct pathway of how it gets there, whether it's DNA, RNA, or RNA viruses that are retrotranscribing. The viral life cycle is a particularly important one because viruses are parasites, and like many parasites out there, they lack the machinery to replicate, and they are 100% reliant on host to replicate and reproduce. There is a little bit of evidence that suggests these viruses can develop a bit, and we can see this here with a micrograph of this Aconitis two-tailed virus. Essentially, it infects the host, it comes out and does some developing over time. But the vast majority of viruses, once they infect the host, once they get the host to make new viruses, they don't change at all. But again, they are dependent on the host, and thus they have to infect a host. Essentially, what they're doing is they insert their genome, whether it's RNA or DNA or any of the combinations of those two, into the cytoplasm of the bacteria or whatever host they're looking at, and this gets incorporated potentially into the host genome if you're one of the retroviruses like HIV. The host replication system then makes more copies of the genome and the proteins. The viral components are then assembled into viruses. And then finally, the viruses are released from the cell via lysis, or what we call controlled release. And so there are two distinct life strategies of viruses, both the lytic and lysogenic cycle. They are very distinct, but they are intertwined with one another. Essentially, the lytic cycle is when the virus begins replicating. As soon as it infects the host, the viral particles get replicated, organized into new viruses, and fully formed viruses break out of the cells via a process called lysis. The lysogenic cycle is what we also call temperate viruses, the idea being the viral DNA is incorporated into the host genome, and therefore every time the cell replicates, the viral DNA is then passed along to the host cell. And it's actually kind of interesting. This is a pretty common thing that viruses do. They By incorporating their genome into the host, the viruses can be passed down for generations and generations. So, for instance, this is how the human genome contains about 7% virus DNA. Once a virus has integrated its DNA into the host, it has then become a prophage. And then the lysogenic cycle does eventually end. Well, it doesn't always have to end, but if it does end, it's essentially some environmental cue and it enters the lytic cycle. So these two are interconnected. I will also mention that this lysogenic cycle is how viruses can transmit DNA from one host to another as well. And I will, just to sort of highlight how the lysogenic cycles work, the best studied case of this is actually the herpes virus. And so if you ever have a cold sore or if you had herpes, that virus initially would have infected you, and its DNA stays in your genome for an extended period of time. And every time you get stressed, essentially the viral DNA or the prophage is activated, causing an infection inside you. So let's break down the lytic cycle in a visual form. So we have a virus attaching to the host. It's adsorbed toward the host and injects the DNA or any nucleic acid inside the host. The protein coat of the virus, that envelope or capsid, remains outside the cell and just sort of flies away. Once the viral DNA enters, it has a synthesis of nucleic acids and proteins that are vital for making the envelope and the capsid of the virus, as well as the genome of the virus. Once that happens, they start to get packaged inside, and then eventually we have the release or the lysis of the cell. The lysogenic cycle is pretty similar. We have a virus attaching, inserting the genome, but the genome gets essentially incorporated into the host genome, becoming a, uh, a prophage, and then eventually over time, cell division occurs, passing down the prophage from generation to generation. Um, as I mentioned, that a prophage can eventually be induced by some environmental condition to then go into the lytic cycle. So they are essentially connected to one another. In addition, we also have one other cycle. It's called the slow release cycle. And so the idea being is we have a virus attaching onto the host. It's a phage particle. It injects its genome. It replicates its genome autonomously, and then what it eventually does, it sort of creates this. The idea being with the slow release cycle, that the host cell continues to grow normally while reproducing parts of the virus, and eventually the phage will, or the virus will essentially lyse the host cell, causing a release. And this is just sort of a nice overall view of what happens in linking both the lytic and lysogenic cycles to one another. It's a nice sort of summary of what we've been talking about the past few slides. But the virus life cycle, in terms of what's happening, the lytic cycle kills the host. The lysogenic cycle allows the host to grow normally, and the slow release cycle eventually allows the host to grow very, very slowly. And it's essentially the idea being is that the environment itself ends up selecting whether the host will enter lysis. So, for instance, when conditions are particularly poor, a microbial growth is slow, virus will typically enter a lytic cycle to get out before growth of the host gets too slow to replicate. When conditions are really good and microbial growth is fast, the virus will enter a lytic cycle to hopefully infect other quickly dividing cells. And when a cell is damaged, for example, by ultraviolet light or heat, the virus will enter a lytic cycle to escape. And when things are just right or okay, it will enter that lysogenic cycle. But again, it ultimately depends on the environment that the virus is in. Because remember, a virus is a parasite. A parasite is 100% dependent on its host. If its host is doing too good or not good enough or sort of okay, it's going to modulate how things are going for itself. And this is just a sort of an illustration to highlight what's going on with our different types of viruses. So we have our naked virus, which is the capsid and the DNA, and then we have our envelope viruses here. And so the process is essentially the same, but what the key difference being is that as the difference between is here is that these our naked viruses essentially escape without it taking any part of the host with them, whereas our envelope viruses, our non-naked viruses, they take a part of the host cell membrane with them. So just to summarize part one, viruses are infectious particles that infect and replicate in host cells using host machinery. They contain a nucleic acid genome and a protein capsid, and this may have an envelope of the host membrane itself. Viruses are classified by their genome, size, shape, and sometimes the hosts and the host range. And the Baltimore classifications, which we dived in a little bit deep into, are based upon genomes. Viruses have typically two main life cycles, lytic and lysogenic. The lytic viruses that begin replicating immediately, they kill the cell, whereas lysogenic ones or temperate viruses incorporate into the genome, then they become lytic depending on the environmental conditions. Part two of viruses, we're going to be looking at what are the stages of the viral life cycle, how do viruses show specificity, and what is the mechanism by which it works and how it affects the host range, and what are the antigenic shifts and shift and drift, and how do they contribute to pathogenicity of viruses. That was a lot. And so this was originally designed to be over 2 lectures, so I'll just skip these few slides. Okay. So the viral replication cycle itself is broken down into 5 stages: adsorption to the host or attachment, delivery of nucleic acids, replication of nucleic acids and proteins using the host machinery, and then assembly of new viral particles, and then finally exit from the host and transmission. So we're going to talk about influenza to talk about this process. It's going to be the model for the day. It's a influenza virus. It's a enveloped RNA virus. And so ultimately what plays the determining role of whether or not a virus can infect a given host is the surface proteins. Um, and whether or not a virus can attach to a host is strictly dependent on the surface proteins found on the virus itself, as well as on the host. They need to match for a virus to physically be able to infect a host. Viruses are limited. A virus's host range is the range of species it is capable of infecting. The host range is determined by specific high affinity interactions between a molecule on the surface of the virus and a molecule on the surface of the host cell. And so because of this, it limits viruses to being able to typically only infect one species, but there are some viruses that have a bit broader spectrum, like say all members of a given family of plants. So for infection of viral capsid, in this case, we're thinking about the naked viruses or the envelope itself must attach to the surface of the host cell. The virus does use the host cell surface molecule for normal cellular function. So for example, we're typically thinking about proteins. So for instance, HIV commonly affects sugar uptake proteins. Binds to sugar uptake proteins on our cells. But can also be for numerous things like iron uptake or potentially conjugation if we're talking about bacteria. Influenza itself uses celiac acid and celiac acid, and bacteriophages use maltose import proteins as binding targets. But it does vary pretty widely. So let's dive a bit into how influenza does its thing. So we're looking at the envelope surface of influenza. We have hemagglutinin and neuraminidase. They have the phospholipid bilayer, and they have matrix proteins. The idea being is that this hemagglutinin in the fluid envelope recognizes and binds to sialic acid on the host membrane. And so sialic acid is a host cell receptor. It looks like this. And it's on the outer membrane of the host itself. And so stage 1, we have attaching. And so idea being we have a gammaphage. So this is a bacteriophage. It has little tail fibers, and these little tail fibers essentially bind to the sites on a protein that is used for transport inside of a bacterial cell. Next up, we have entry and uncoding. So the first steps in a viral life cycles involves attachment and uncoding. In most animal viruses, unlike bacteriophage, they do enter the host cell as virons. So um, physical just pieces of DNA. In this figure itself, the virus has an envelope, but similar processes also occur in other viruses. There are two main ways that viruses enter the host cell. Some viruses attach to the host cell receptor, molecular they recognize, so here. And then they fuse with the host cell cytoplasmic membrane and introduce their nucleoprotein core into the host cell. Um, for other viruses, the binding of the viral protein to the host receptor is stable enough to trigger the cell to initiate endocytosis by the virus. Um, Endocytosis is a normal pathway used by mammalian cells to ingest nutrients, and the virus takes advantage of this pathway. The idea being is we have a virus like hepatitis C, it binds to the protein, the cell itself engulfs by endocytosis and creates an endosome around the virus. It fuses with the virus and it acidifies it. But ultimately, after that endocytic vesicle is formed, it fuses with the lysosome and it begins to acidify. Acidification leads to conformational changes, so just simple changes in structure in the matrix proteins that underlie the envelope itself. So right here. It also affects the envelope proteins, which mediate the fusion of the host and viral membrane so that the nucleoprotein core of the virus is released into the cytoplasm. And then ultimately we have uncoding um, of the viral genome itself. For something like the Zika virus, that's a mosquito-borne pathogen, the virus does enter the body through endocytosis and then co-opts the cell's machinery to replicate its RNA, translate it into a protein, and then is released via exocytosis, which is the reverse process of endocytosis. Um, in terms of influenza itself, the way influenza gets out of a vesicle is particularly interesting. The influenza protein is interesting. The influenza virus, I'm sorry, is interesting because they have these series of fusion proteins. The idea being these fusion proteins are normally buried inside the hemagglutinin trimmer as they are hydrophobic. Idea being is that they're sort of buried inside the cell itself because they're afraid of water. But ultimately, after the lysosome itself fuses with the vesicle, the protons that flow into the viral particle itself through a protein channel called M2, which is right here. And then the acid changes the conformation of the fusion proteins in a way to poke a hole of the vesicle in the membrane, releasing the viruses itself into the cell and eventually into the genome. Phage entry is a little bit different, but not so different than you would expect. They do attach to specific receptors on the cell surface of the bacteria they infect. However, they do not attach to that salic acid, which most human-based viruses do. However, they attach to proteins, and in particular, they attach to sugar transport proteins. Sugar transport proteins are pretty common across all domains of life, so they're pretty common targets for most viruses. Phages themselves also have a specialized tail that will clamp onto the host cell, and the tail fibers bind to the proteins and the sugars on the cell surface itself. And then eventually the base plate, which is the sort of bottom part here, makes contact. And then the contact in the base plate generates essentially this change in the sheath, um, which causes a widening and shortening in a way that injects the tube straight through the host cell wall and the cell membrane into the bacteria. And then eventually the genome is injected into the host. So it's actually kind of interesting. It behaves a bit like a hypodermic needle in this case. If you have time, there's some pretty fantastic videos of this process occurring. Next up is replication. So RNA viruses replicate in cytoplasm themselves, while DNA viruses replicate in the nucleus. Influenza is an exception. It's an RNA virus that replicates in the nucleus, so it sort of bucks the trend. Usually gets out of the nucleus. Usually getting in and out of a nucleus is a sort of a tightly controlled thing. Cells don't like to let a lot of things leave and enter a nucleus. but the virus does have some tricks itself to mimic the host proteins to get through nuclear membrane pores. Once the the idea being is we need to make messenger RNA that can be translated into proteins, and again, we need that positive sense or the top strand of the RNA. And so the type of mRNA can be typically translated through the host. It typically transcribed via host ribosomes, and it creates a plus strand of mRNA due to the orientation of the ribosome itself. Again, it depends on the genome of the virus, but translation can typically typically does need to occur, or does go through some intermediate steps. But remember, during replication, the genetic material itself has two main purposes: to serve as a template to make proteins and serve as a template to make new genetic material for viruses. And so just as a sort of a way to illustrate what's going on here, we're going to talk about a single-stranded RNA virus. In this case, we're talking about polio, that disease that is sort of eradicated, but not so much because of the anti-vaccination movement. But we have this plus strand of the parental RNA. It's then translated by reverse replicase, and then capsid and envelope proteins are made. And then we create a negative strand, and then we have a replicase create a positive strand, and then they have capsid and envelope proteins form, and then we create a progeny virus. So we sort of go from positive to negative to then do all that we need to do, and then it goes back to positive, and then back to goes back to positive to make more viruses. Retroviruses are a bit different. They make plus-stranded parental RNA. They have an enzyme called reverse transcriptase to make a copy of single-stranded DNA. This is then translated into double-stranded DNA, which then integrates into the host genome, and then the host RNA polymerase is used to make all the required RNA for new viruses, or an RNA to make the proteins itself to make more viruses. DNA viruses are particularly are simpler. They have the double-stranded DNA. The host virus DNA, the host or viral DNA polymerase is used to make more double-stranded DNA that is essentially used to make more copies of the viral genome for the progeny. But the host DNA polymerase, as well as RNA polymerase, is used to make proteins and mRNA needed to build more viruses. And retroviruses include viruses like HIV, and double-stranded viruses include those like herpes. Next up, we have assembly and departure. The idea being is we need to assemble our viruses. Once we make the genome itself, and once we make all the proteins, we need to assemble it into a virus. And so when the concentrations of viral genomic fragments and viral proteins reach sufficiently high concentrations in the cytoplasm of the infected cell, the proteins from the nuclear protein core start to interact with the viral genome segments. Eventually, the viral particles begin to bud out through the cytoplasmic membrane, and then they're acquiring the envelope in the process. At this point, there's one final problem. The hemagglutinin protein spikes that stick the virus to the cell in the first place still want to bind to the sialic acid residues on the mammalian cell surface. If they do, the virus would get tethered to the cell that is trying to leave. However, we have this other protein, neuraminidase, that cleaves the bond attaching the salic acid residues to the host cell of the protein. The idea being is that this protein itself would want to continuously bind to the host, but this secondary protein here, which I'm not even going to say, prevents that from happening. So what we would normally see is these binding to one another, preventing the virus from leaving, but this secondary protein prevents this. But ultimately, remember, it needs to sort of get away from the host to then go on infect. Because remember, a virus's whole purpose is to simply infect other cells. And so we've been sort of talking about influenza a bit. There are three main types of influenza, A, B, and C, which differ in their amino acid sequences of their protein envelope proteins. The influenza typically causes several million cases a year across the US, and it's estimated to cause typically about 20,000 deaths a year, but it does vary pretty does vary pretty variably from year to year. So this particular year was a pretty bad year. The previous year was not so bad. Um, Due to the sialic receptor, it can attack multiple different types of human cell. So it doesn't have to just infect, say, lung or tissue. It can affect brain. It can really affect anything, simply because the sialic acid receptor is pretty common to all of our cells, regardless of whether they are in the body. It is a fast evolving RNA genome itself. Um, and it does undergo consistent changes in its envelope proteins. And it has antigenic drifts, so essentially small changes in the envelope proteins, versus antigenic shifts or major changes in the genome. Um, and so when we have minor shifts in the envelope proteins, not too much happens. But when we have major shifts in the genome of the viruses, this is where we get major killer flus. And so when people pass away from the influenza itself, you typically don't die from the influenza virus. You typically die from a subsequent infection by a bacterial pneumonia. It can also be viral as well. You could also, in turn, get COVID-19 if you, say, had influenza as well. Influenza virus himself attacked epithelial, sorry, epithelial cells in the respiratory tract. And these tracts are mostly our first line, or not mostly, they are a first line of defense in our lungs. So they do weaken sort of our first line of defense, allowing subsequent infections like bacterial pneumonia. There have been many, many epidemics of influenza. However, I'm sure you all have probably seen this in the news at some point, given how much screen time COVID-19 is getting. But in 1918, we did have a pretty major outbreak of influenza. It killed between 50 and 100 million people, which at the time was, 5 to 10% of the population. Um, um, Influenza as a whole is particularly easily treated with vaccines, provided you get them. And influenza itself is particularly less deadly to the young and more so to the old. However, flu vaccines are basically have to be made every year, simply because the virus mutates particularly quickly. And it's in the primary thing we actually see is what we call antigenic drifts. What we're seeing is antigenic drift, is the idea being is that year to year, the changes in the envelope proteins that are small or sometimes large essentially make it so we have to develop a new flu vaccine every year. But the evolution of killer flu is a sort of an interesting one. So the two big changes that have occurred because of the influenza include its segmented genome, as well as the ability of influenza A viruses to infect mammals and birds. And so an example with the pig we have here, although you probably have heard of the main new scare of influenza itself as birds. But suppose the pig is infected with simultaneously with both human influenza as well as influenza from another species. In this co-infection, the two types of viruses replicate in the same cell. Predominantly the human virus can pick up the genome segment from the porcine or pig virus. This may result in a new strain of the virus with new envelope proteins that won't be recognized by the human immune system. Um, this is, when you see things like bird flu or swine flu, this is what's happening. One of our viruses is infecting another host and is picking up something from another host itself. So essentially, we're creating, I guess, killer strains by our sort of close association with a small subset of animals. Influenza in pandemics. So as we mentioned, the last major flu was in 1918. It hit so deadly because it was the majority of the proteins themselves were actually derived from bird influenza. So this really deadly pandemic that occurred in 1918, H1N1, which I'm sure you all heard was a recent scare a few years ago. And the last time we had a sort of a major pandemic response in the United States was because of H1N1. The idea being is that these extra proteins that we find in bird influenza or, say, pig influenzas are not particularly well recognized in our own host by our own host immune system, simply because we're not generally exposed to them. But as we continuously expose ourselves to these, it is likely we will build up some immunity, but it's also likely that as we expose ourselves more and more, we're likely to find new and deadly combinations that will lead to future pandemics going forward. Um, but just as a sort of a summary of what's happening, here's a nice table of sort of the five major things that are happening, both influenza as well as other virus strategies for your own personal view. So just to summarize part two, there are five stages of viral replication: attachment, uncoding, replication, assembly, and exit. Specificity of viruses comes from the fact that the receptors um, attach to the specific targets on the host cell, and this does create a limitation in the types of hosts that can infect, as well as the host range. Though, as you saw with the example of the influenza, we're not particularly closely related to birds, but birds can be infected by our influenza. Because one cell can be infected by many viruses and their genomes can be packaged randomly, viruses can have particularly dramatic shifts in their genomes, which may make them very deadly. So that's part two. Part three, we're going to talk about viral genomes. So what are the types of viral genomes? What types of genes do viruses carry? How do viral genomes evolve? We're going to talk about HIV as an example, because it's, outside of COVID-19 is the consistent pandemic on this planet. And then what can we infer about life from viral genomes? So what do viral genomes carry? The short answer is not very much, mostly just proteins to make new viruses. But if you remember back to our horizontal gene transfer class, they can carry special stuff. So remember, and we're going to talk about this with the idea of the Prochlorococcus phages. So Prochlorococcus is one of the most abundant microbes on the planet. They're autotrophic. You find them in the ocean, and they are incredibly important to how the Earth produces oxygen on a day-to-day basis. They're phages of themselves that infect Prochlorococcus. So we're seeing Prochlorococcus. We're seeing a Prochlorococcus phage here. What ends up happening is their phages have been shown to pick up a complete suite of photosynthetic genes, meaning that while these phages themselves cannot photosynthesize, they can then transmit those photosynthetic genes to a completely unrelated microbe. It's a particularly wacky thing that we see, but again, most viral genomes don't carry very much, but they can carry some wacky and wild things. If you're interested in this sort of idea, this paper here, published in the Proceedings of the National Academy of Science, is a particularly interesting read to see some of the wacky and wild things phages can carry. But viral genomes, they have a vast diversity of shapes and sizes, as we've talked about. And so just to sort of highlight some of these crazy and wild shapes, ranging from the bacteriophage to Ebola to HIV to cowpox. They have a pretty wild and wide diversity of sizes, shapes, genomes. Um, there is some estimates that say 20 to 60 viruses per mammalian cell. That estimate is a bit low. Most estimates will put it at 1,000, which is pretty crazy. There are, if we sort of extrapolate from that, there's 100 million species of viruses out there, provided there are 1.7 million known species of vertebrates, invertebrates, plants, lichen, mushrooms, and algae. So the take-home message is their viral genomes may hold the greatest of all gene diversity of life. It's simply an undescribed diversity thus far. Just as a reminder, viral genomes are separated by DNA and RNA. They can be linear, circular, double-stranded, single-stranded, so on and so forth. They can also, remember, be plus sense, minus sense, or plus minus sense. It's a whole wacky and wild thing for viral genomes. The next question we can ask is, are all viral genomes smaller than bacteria? And so the largest virus that we know of is about 0.4 microns. The smallest is about 0.01 microns. And the typical bacteria itself is 1 to 5 microns. But what you'll notice is that the size of viral genomes is really small, 9.2 kilobases. So that's 9,200 bases long. And then if we sort of go up the ladder, looking at, say, something like West Nile virus, SARS, a T7, or a gamma phage, which are bacteriophages, they're pretty small, right? 48,000 bases of DNA or RNA long. Um, However, there are some viruses, including the Mimi virus, which we'll talk about in a minute, that have particularly large genomes, 1.2 megabases, which is about the size of a bacterial genome. And so we can look at genome sizes of, so this is genome size in the bottom, and base pairs. We have, and it goes up as we go from left to right here. What you'll notice is that mammals, plants, fungi, and yeast, they're big. Bacteria, much smaller. Viruses, even smaller. However, there are some what we call giant viruses. So as you'll notice here, we have the Mimi virus, which does have a bacterial sized genome itself. And so the Mimi virus is a really interesting and one of the sort of the newer surprises from microbiology looking at nature. So it's a virus that looks like this. It's kind of like a hairy hexagon, but it infects this a protist called Acanthamoeba polyphagia. This phage occurs in was found to occur in hospital environments. A French group sequenced its genome and annotated with 1,200 potential genes. It had one linear chromosome with reverted repeats, meaning that was likely one circular. Um, and this caused a major sort of shift in the virus world. It caused a lot of people to question what we knew about viruses. It caused a lot of people to examine what we knew about viruses or potentially rewriting some of the rules about viruses because it is a monster virus. Again, it's bigger than some bacteria. It has a genome bigger than some bacteria. It is huge. It is not typical of what we see of most viruses. Um, so Acanthamoeba itself is a pathogen. So it is found in unprocessed Acanthamoeba being not the virus, but the the microeukaryotic parasite. It is found in unprocessed and domestic tap water, ice, fresh lakes, seawater, swimming pools, hot tubs, insects like cockroaches, soil dust, and sewage. It can cause lung infections inside of human hosts. Um, itself. But it does have this virus called the Mimi virus. It has a zero point 0.3 microns wide, which is about the size of some tiny bacteria. So to put that in perspective, when you guys were looking at Staphylococcus in the lab, it is approximately 0.5 microns wide or micrometers wide. So it is a pretty hefty virus. It has no cytoplasmic membrane. It has no cell wall. It has no metabolic processes, but it has a core itself. It has a double-stranded DNA virus. It has 90% coding capacity, meaning that of the entire genome, 90% of it codes for proteins, which is about what we see for most bacteria. It has about 1.2 million bases, and it has anywhere from 900 to 1,200 base genes in it. And it's huge. It's capsulated, it has an inner membrane, and has that core. When we look at its genome, it has a very bacteria-like genome. It has an origin of replication. It has GC content of 28%, which is very similar to most bacteria. Just as a reminder, GC content is simply what percentage of the genome is guanine and cytosine as opposed to adenine and thymine. Um, and it's very bacterial-like. It has the origin of replication. It has genes that are similar to bacteria. And most viruses themselves need a host to provide replication, translation, transcription. The Mimi virus actually doesn't. It is capable of autonomous replication if it wanted to, but it simply actually doesn't. And we're not quite sure why at this point. In terms of viruses themselves, viruses are classified as being non-living simply because they don't do many of the things that we characterize most living things to do, whether they're bacteria or they're humans or yeast. Viruses simply just don't have many of the characteristics of life itself. The Mimi virus, when you look at it on a phylogenetic tree, it's actually particularly very similar to both archaea and eukaryotes. And so it seems to be that Mimi virus is one of these falls into the reversion hypothesis where we had a parasitic either eukaryote or archaea, and it reverted to being a virus. And so it's sort of this oddball where it's most viruses are sort of off on their own here, not related to anything, but this one seems to be more closely related to archaea and eukaryotes. So it's a particularly wacky and wild thing. Um, and so some people have sort of questioned, is Mimi virus, given the fact that it can replicate by itself, is it alive unlike other viruses? And the answer to that question is, I don't know. No one really kind of knows. We think it might have been a parasitic eukaryote that lost its genes to look like a virus, but it really does blur the distinction between prokaryote and virus and eukaryote and virus. That being said, Mimi virus is not the only large virus. There is a group called the megavirales. You take a peek at some of the viruses there. They are known to not really infect us. They're known to infect human. They're known to infect like amoebas and micro eukaryotes. So nothing for us to worry about at the moment, which is, I think, good because we probably have enough to worry about at the moment with viruses. So moving on from the oddball of the virus world, we're going to talk about how viruses evolve, and we're going to do this through the lens of HIV, or the human immunodeficiency virus. And HIV is a particularly simple virus. It's got its capsid, and it's got its enveloped. So again, this envelope is going to be from the host itself. It's got its enzymes, and it's an RNA virus. And remember, HIV is a retrovirus, meaning it requires its RNA to be reverse transcribed to DNA to do anything inside the host. HIV itself targets T4 lymphocytes, so these are a part of your immune system involved in the adaptive response to new pathogens within inside you. HIV has, like most viruses, has that similar 5-step process of binding, fusion, uncoating, assembly, budding, and maturation. And so it binds onto the CD4 receptor. This is a sugar receptor on the outside of all human cells. But it's particularly pretty abundant on T4 lymphocytes. It binds to this protein, it fuses, it uncoats, again by attaching onto one of those lysozymes to break down the coat. A reverse transcription occurs, creating a viral piece of DNA that's incorporated into the host, and then the host is then reprogrammed to making viral RNA, making viral proteins, making a genome, assembling all this stuff, and then budding off, and eventually fully maturating once it leaves and detaches from the cell. So that's HIV. Pretty simple, pretty consistent what we've been talking about thus far. Nothing crazy outside of the reverse transcription that has to occur. And so HIV, in terms of as a pathogen, it involves depletion of T cells by disrupting the plasma membrane and the permeability of those T cells. It causes the destruction of immune cells due to the presence of GP120 in the membrane. GP120 is simply a protein that is involved in the structural stability of our membranes, and it ultimately can also cause apoptosis or programmed cell death. It does cause a disruption of the balance between different T cell populations. So T cell populations inside your cell are important for how you mount your immune response. It also might disable or destroy dendritic cells, so cells that are involved in your nervous system. That being said, HIV does mutate extremely rapidly, so it evades the host immune system pretty effectively. So, for instance, there are about 10 to the 10 viral particles produced each day by HIV, and there are about 3 times 10 to the 5 times 10 to the minus 5 errors per nucleotide. So every 3 times 10 to the 5 nucleotides that are synthesized by HIV, there is an error in the genome itself. So it mutates fast. This is much, much faster than anything else we know of mutates. But retroviruses as a whole, they are single-strand RNA viruses. They are known to cause tumors in chicken and mammals. But they are particularly infectious in terms of HIV. And so the way this works for retroviruses, we have the viral RNA. We have an enzyme called a reverse transcriptase, which converts that RNA into viral DNA. And once we have that viral DNA, it then incorporates itself into the host genome as a provirus or a prophage. Um, Reverse transcriptase is a really interesting enzyme because it lacks a proofreading function. We didn't really talk about this in depth when we talked about how bacteria replicate their genomes, but when things replicate their genomes, they're very, very accurate. You, as a human, have an error 1 in every 10 billion bases. Bacteria have an error 1 in every 5 billion bases. Um, as you saw, HIV, this enzyme, every time it makes a copy of the viral genome, has an error 1 in every 30,000 bases. And it does this simply because it doesn't proofread. It's very tolerant of incorrect base incorporation. Therefore, HIV, as with all retroviruses, because of this reverse transcriptase, have a very high mutation rate. And this is why HIV is particularly problematic to deal with. In terms of what the genome of HIV has, it's a very small genome. It's only about 9,700 bases long. The key enzymes here is Pol, and this is the reverse transcriptase. We have Pro, which is a protease. We have ENV, which is a glycoprotein. We have GAG, which are viral proteins to make the matrix, the capsid, and the nucleic acid binding proteins. And then we have the SERC or the ONC genes, which are necessary for transformation, but not replication. And so once you have HIV for long enough, you progress into AIDS, which is short for acquired immunodeficiency symptoms, and this happens due to the progressive destruction of CD4 cells, leading to the collapse of immune systems. Patients that have AIDS are much more susceptible to opportunistic infections, including yeast infections from a yeast called Candida. It's also more susceptible to invasive cervical cancers if you're a woman, because most cervical cancers are caused by viruses, and a whole number of different infections and cancers. People that have AIDS are much more susceptible to. And so we can look at how this sort of progresses over time. So we have the CD4 lymphocyte account and in blue here, and we have the HIV RNA copies per milliliter of plasmid. Then we have weeks at the very first part and then years at the later part. So what you'll notice is that we have that primary infection that occurs here. And as the HIV builds its numbers over time, we see a pretty steady tanking of the CD4 cells inside your bloodstream. After sort of this acute portion of HIV syndrome, HIV is then sort of circulated through your blood system, and in particular, it seeds inside your lymphatic system, so sort of the dumping grounds of your immune system. As things progress, HIV population does dip down, and your immune system does recover a bit. But over time, we start to see your CD4 cells, simply because they're in this clinical latency phase, sort of dip down over time, eventually hitting zero, because HIV will eventually wipe out your entire CD4 lymphocyte count. But you'll notice is that HIV sort of dips down and sort of eventually it sort of slowly increases its abundance over time. And as it starts to get particularly bad and your immune system is heading down to a zero, we're seeing sort of late stage, we have AIDS forming here, but you'll notice AIDS is classified by very high amounts of HIV in the blood, as well as many, many opportunistic pathogens and eventually death. People that have AIDS do not die from AIDS itself. You theoretically can live with without immune system, but you typically die of an opportunistic pathogen. Most common are parasites, as well as respiratory infections. HIV in itself is classified as a lentivirus. It was first described as the third human retrovirus. There are two distinct groups of HIV, which you'll see in a moment. HIV is incredibly diverse. So HIV-1 is the dominant worldwide strain of HIV. This is what we have here. This is what we have in China. This is what we have everywhere else. Then we have HIV-2. It's less virulent, meaning it is less deadly to the host, and it's endemic typically to Western Africa. But what's interesting about these two viruses is they have two distinct inferred origins. So HIV-1 we know came from the common chimpanzee. HIV-2 we know came from the sooty mangbe. This is the sooty mangbe, and this is obviously a chimpanzee. And so HIV was sort of got not sort of, did gain a lot of traction in the 80s. It was sort of this new disease. We had no idea what was going on, but we do believe now that HIV has been hanging around since before the 1950s in Africa. Uh, Globally, there are about 40 million people or so living with HIV worldwide. About 1 million people die every year of HIV-related diseases. So it's up there in terms of causes of death. And it's not a top 10 cause of death, but it is a particularly damning cause of death. But what you'll notice is this map from the World Health Organization. As you'll notice, the vast majority of HIV cases do occur in Africa, but however, the Americas are particularly uh, chock full of people with HIV. Just as a reminder, HIV is a sexually transmitted disease, and so it's transmitted either through um, sexual interaction, so the exchanging of body fluids in that case, unprotected sex, as well as through blood transfusions from infected person to non-infected person. That is the only mechanism you can get HIV. You can't get it from a mosquito. You can't get it from a toilet seat. It simply has to be through the direct transmission of bodily fluids. But as you notice, is that people can be asymptomatic of HIV and not really know they have it and still spread the virus. Um, and so, as we mentioned, HIV has two groups. We have HIV-1 and HIV-2. HIV-1 is the global one. HIV-2 is the African endemic one. And what you'll notice is that we have HIV group 1 here, and in that HIV group 1 is actually divided into a number of different subgroups, M, O, and N. And what you'll notice is that it's actually closely related to the CPZ, or the chimpanzee immunodeficiency virus itself. That's in contrast to HIV-2, which has a bunch of different classifications, 2B, 2A, and SMM, SIV, SMM. And you'll notice that it's closely related to the SIV, or the simian immunodeficiency virus. And what's actually sort of interesting about these and what we can infer is that it seems to me that there was likely multiple jumps for HIV-1 to make it to the population. And so HIV-1 seems to have jumped 3 distinct times, leading to these 3 distinct subgroups here. So we have HIV-1, O, N, and M. These are likely 3 distinct events from HIV jumping from a chimpanzee to a human. That's in contrast to HIV-2, which seems to only have 1 inferred origin, and only jumped from a simian once strictly to us. And I just want to highlight one thing. When I say jump from an animal, and given the nature of HIV, this is not like it jumped because somebody was having sex with some sort of primate. This is this is through the consumption of bushmeat. This is repeated exposure to raw meats that led to the absorption of this virus into the human through like an open wound, either in the hand or the mouth. So this is not like, you know, something gross with someone having sex with a chimpanzee. This is just bushmeat, exposure to bushmeat, and this is particularly this is particularly relevant now because this is how we believe that the current COVID-19 epidemic is actually made the jump from bats to humans as well. So humans, animals are a really good reservoir for humans. And so new infections we believe to have occurred before the 1950s in Africa. So here we have HIV-2 lineages in green, HIV-1 lineages in red, and all other SIV lineages are blue. And the question we can ask is, what does this suggest? And if you want to sort of take a deep dive into this, I recommend taking a peek of this at this paper. It's a really interesting paper. But what it does suggest is there are relatively this is given, and I'm sorry, I should mention that this is the sort of the wild version of the simian influenza virus. And what this does suggest is this was likely an old virus, likely that HIV-1 and 2, like HIV-2 likely existed in the population prior to modern times, whereas HIV-1 is a much more modern introduction, likely happening in the 80s or so. And sort of the one last thing about the evolution of HIV, it does evolve very rapidly, and the idea being is if I had HIV and I gave it to someone else, within a few months we would actually see some particularly dramatic changes in the HIV genome, and that's exactly what we see here. So this is just a phylogenetic tree. Each one of these colors corresponds to the um, HIV population found within a single individual. So this is person A, B, C, and so on and so forth. And what you're seeing is that even within a single host, there's a vast diversity of viruses. But what you'll notice is that the HIV found in one person versus another person is completely distinct. So if you get HIV from someone within a few months, you will have your own distinct and unique population of HIV due to the rapid evolution of HIV. And so, just as sort of explanation of how this works, HIV has a few minor contributions in terms of how fast it evolves. It does have a high rate of viral replication. It can produce 10 to the 10 viruses per day, which is much higher than most viruses. Most viruses produce 10 to the 6, 10 to the 7 per day. HIV produces a lot of viruses. The size of the viral population in individual patients can be particularly very large. Um, and remember, it can have extensive recombination. The idea being when you have two two viruses infecting the same cells, you can have a cross-linking of the viral genomes, very much like what we see for influenza. But the major contributions to this are that essentially there are mutations that occur during retroviral replication. So as a result of this reverse transcriptase enzyme, remember it has no proofreading, thus it has a high error rate. What this does is it allows HIV-1 to evade the host immune system. It alters what's called tropism, which is basically the type of cells viruses can infect. So it allows these to infect multiple different types of cells within your immune system, and it also allows the virus to develop resistance to antiretroviral drugs. So the take-home messages from this part is that the lack of fidelity in the replication does contribute to the adaptability and fitness of the viruses. Retroviruses themselves, like HIV, have some of the highest mutation rates known on the planet itself. And this allows HIV to evolve very rapidly. That being said, there is some evidence to suggest that HIV might be evolving to be a less virulent form. And so remember, all viruses are parasitic. A good parasite is good at infecting its host, but not so good at killing its host. Because remember, a virus needs its host to survive. And so if a virus is too good at killing its host, it will essentially deplete its population, so it will no longer be able to spread effectively between hosts. So it needs to strike a balance between how effective it is at killing the host and how effective it is at sort of keeping the host alive. And so there is some evidence to suggest that within HIV-1, there's this new group developing here. And these new groups are potentially less pathogenic. So remember, HIV is a particularly new virus. It was only something we've been dealing with globally, pandemically, for about 30 to 40 years now. And it's only been around for less than 100 years at this point. So it is something that's going to evolve and change over time. And we, as a human population, are also going to evolve and change over time in response to HIV. This is in contrast to something like measles, which we've been dealing with as a human population for about 10,000 years. And measles is something that is deadly, but not too deadly itself. And so just to summarize the third part of this, viruses do have small genomes with no metabolic capacity. Remember, they are strictly reliant on their hosts. We talked about the curious case of the Mimi viruses. They're bacterial-like, but they resemble eukaryote phylogenetically. They really do blur the line between living and not living. HIV itself does evolve rapidly, and this is due to its faulty reverse transcriptase enzyme. And if you want to look at some cool, fun stuff viruses do, I'd recommend taking a peek at this website. It's really interesting. Viruses are a really crazy group of organisms, and there's so much we're learning about these viruses every day, and it's really crazy what they can do. And so just to wrap up, we're actually going to finish by talking about a process inside bacteria and archaea that we have adopted to be something that is clinical in nature, but it is directly related to viral infection. And this is the CRISPR-Cas9 system. So the questions we're going to answer today about this are: what is it, how does it work, and how can we use it to make our lives better? And so CRISPR-Cas is an adapted immunity found inside of bacteria as well as archaea. CRISPR stands for clustered, regularly interspaced, short palindromic repeats, and we're just going to call it CRISPR because that's a mouthful. I have a friend that researches CRISPR, and she can't even remember what it says half the time, what CRISPR stands for half the time. So we're just going to call it CRISPR. But it is an adaptive immune system for bacteria and archaea. It's found in 90% of archaea and about 50% of bacteria. And so you notice in the title, there's palindromes. A palindrome is something that is the same forward as it is backwards. So taco cat is the same forward and backwards. A man, a plan, a canal, Panama is the same backwards and forwards, and so on. My favorite is doc note, I dissent. A fast never prevents a fatness, I diet on cod, which is the same forward and backwards. And so we're talking about palindromes because these are palindromic. And so palindromes, again, same forward and backwards. And so CRISPR has three major components. So they have the Cas genes, they have the leader sequence, and the repeat spacer array sequences. And the idea being how this all works, we have a phage infecting our bacteria or archaea. It injects its DNA. What normally happens is, if you remember, the phage will essentially its genome will go, incorporate into the bacterial genome, become a prophage, and then reprogram the bacteria to make more phages. What ends up happening in our CRISPR-Cas9 system is the double-stranded viral DNA is bound by these Cas proteins, and what it does is creates a novel spacer. Then what happens, once you have that novel spacer, what ends up happening is upon a subsequent infection, the viral DNA is bound by Cas proteins and then is thus inactivated. And the idea being is that once you inactivate the viral DNA, you essentially can't, the virus DNA can essentially not reprogram the host. But the idea being is after you create a novel spacer, those novel spacer regions are used to essentially tack onto Cas proteins to have a receptor to then bind to the viral DNA. And so each one of these is in this case here is specific to a specific type of virus. And this is again, it's something that is adapted. It's very much like our B cells, which are our memory cells that allow us to remember something that we were infected with. And this allows a bacteria to remember what it was infected with. And the idea being, and so the idea being is that these bacteria can use CRISPR and these Cas proteins to essentially have their own immune system. Again, it's immune against viruses. That's really their only, well, not their only, but their major predator is evading viruses. That being said, we've developed CRISPR actually to be a biotechnological thing. So I'm sure many of you saw last year that there's this whole big controversy coming out of China about this technology of CRISPR being used to edit the DNA of a human embryo. And so it is has been, and so we've adapted it to use CRISPR to make a technology, a biotechnology to edit genomes. And so if you want to take a look at this review paper, it's a really good overview of sort of the things I just basically touched upon. But the idea being, we have had a number of things throughout history to edit genomes, but CRISPR-Cas is a really powerful tool relative to other things because it is programmable, it is very easy and cheap to do. You all could do it in the lab. It's really easy, and it's a very specific target recognition. Because remember, CRISPR is based upon the sequences that are found within that repeat region. And so the idea being is we can use CRISPR to essentially break apart a piece of DNA, insert a new or delete a part of the DNA, and then splice it back together. Essentially, we can edit genomes, whether it's eukaryotic or prokaryotic or even viral, on the fly. And it's really something that has shown a lot of promise in terms of editing editing genomes. And so, for instance, as I mentioned, there was sort of this controversy of these children in China. What these researchers in China did was use CRISPR to actually edit their genome to make these infants not susceptible to HIV. So they would no longer produce the protein that HIV would attack. Um, and so CRISPR itself has the potential to revolutionize biological research and affect personalized medicine. And it's a really powerful tool. Remember, it's easy, it's very targeted because remember, it requires that one specific sequence. And it might be something that's really powerful to correct genetic mutations, and it also has the potential to change how we develop our crops in terms of making better, more drought-resistant crops or disease-resistant crops. Again, we might be able to improve this so that it's sort of much more accurate at fixing, say, simple or potentially complex issues within human genomes itself. Um, but as with all biotechnology, there are some issues. The good could cure diseases, makes things better in terms of plants and animals. It can also make essentially uh, retrovirus-free pig organs for transplants. The not so good is eugenics, so making designer babies, superhumans, etc. It also might be something that might lead to bioweapons, or it might be some of this concept of racism or classism, simply because these technologies right now are really only for people that can afford them because they're not really covered by like insurance or things like that. And so it might create this really not so fun system of racism and classism, which clearly already exists, but could potentially be exacerbated by this crazy technology. And it's also something we should potentially fear because we don't know quite what's going to happen. And that's sort of the issues people had with what happened in China. We're not really sure what are going to be the long-term effects of the editing that has occurred here. And we simply just don't, even though we've been studying the human genome for a very long time, we still don't know enough to accurately make predictions of what's going to happen when we edit one specific region of the genome. Um, but CRISPR itself is unlike anything that we've ever seen before. And is going to be something that's going to change future generations. Right now, it's in its infancy. And one particular way we think we're going to be doing this is to say circumventing the possibility of inheriting a gene through this process called a gene drive. And the idea with the gene drive is if we have, say, a CRISPR edited um, organism versus a non-CRISPR edited organism, the idea we can use CRISPR to convert a gene inside this organism to be a beneficial gene or say a less detrimental gene. And then what we can do is over time, by breeding it, trying to eliminate these sort of bad genes from the population, and allow these genes to sort of these CRISPR edited genes to sort of flow through the population by the introduction of these new CRISPR edited genes and mating. The idea being is that potentially we could alter the genomes of, say, mosquitoes to, say, make them not be able to carry malaria or dengue or yellow fever or any of the other nasty diseases that mosquitoes carry. And this could be something that could be game changing because malaria alone kills more than 500,000 people every year. The vast majority of them are children. Malaria is the most common infectious disease on the planet at this point, outside of like colds and influenza. It sickens more than 200 million people every year, and is an extremely costly thing, both to not only just Africa itself, but also to those that are trying to help the African people. We also think we might be able to use gene drive to eradicate invasive species. So species that are, say, usually only found in, say, China, but come here, or species that are, say, found in only in New England, but they're transported to, say, Nepal, something like that. So we might be able to use CRISPR to edit the genomes to potentially eliminate invasive things. It also might create sustainable agriculture by essentially reusing, eliminating the need to use pesticides or making pesticides more efficacious. And so just to summarize this part about CRISPR, so CRISPR is an adaptive immune system for protecting bacteria and archaea from viral infection. The CRISPR itself is a powerful genome and gene editing technique with the potential to change the face of the planet, but there are lots of pros and cons to that, very much like all biotechnology as a whole. And so with that, that's going to be the end of our lecture today. Please let me know if you have any questions. The slides will always be up on my RCC, but please do let me know if you have any questions about the material. I'd be more than happy to discuss them either like on Skype or by the phone or simply by email. And thank you very much. I hope you all are doing well and are being safe. Take care.